Good morning. Thank you for joining us today for our MDICx webinar, a case study in how patient preference information contributes to regulatory decisions for medical devices. My name is Stephanie Christopher. I am the MDIC Program Director for our Science of Patient Input Initiative, and it's my pleasure to be able to welcome you to this webinar and to welcome our speakers. We are especially excited to be talking about this topic of how patient preference information can contribute to regulatory decision-making. Many of you may be familiar, MDIC has worked hard in this space of informing the science of patient preference information, and we're delighted to be able to share this successful case study with you. Our speakers today include Martin Howe, the Associate Director for Quantitative Innovation at CDRH, Frank Hurst, the Medical Officer in the Division of Reproductive, Gastro-Renal, and Urological Devices at CDRH, and Todd Snell, Senior Vice President for Quality Assurance, Regulatory, and Clinical Affairs at NxStage Medical. We've reserved the last 10 to 15 minutes of our webinar today for questions. If you have questions, you can submit them via the chat box, which is a feature on the column next to on the WebEx screen. So with that, I will turn it over to Todd.
Great, thank you. Good morning. Again, as Stephanie mentioned, I'm Todd Snell, representing NxStage Medical. For those that don't know us, we're a small, mid-sized company based in the Boston area. We were established in 1998, and our primary focus is really renal disease, chronic kidney disease in the home, acute kidney failure in critical care environment, and also we produced disposables for an in-center dialysis environment. And over the past few years, we've actually opened up also our own NxStage kidney care dialysis clinics. Really excited to speak today about how did we get to a solo home hemodialysis indication. I'm going to talk a little bit about why solo is important to us, why patient preference was also important in a really innovative pathway. I'm going to talk a little bit about how the pathway evolved, and then give some challenges, lessons learned, and some advice for other sponsors that are possibly considering a patient preference pathway. Next slide, please. So for those that don't know, from a NxStage perspective, solo home hemodialysis is really the ability for a patient to basically cleanse their blood, remove toxins and additional fluid while they're in a home environment without the support of an observer. So a little bit of background, our original home hemodialysis indication was in 2005, and specifically in the labeling called out for an observer to monitor and observe treatment. And back in the day, I would assume the renal branch and NxStage Medical, as this new modality for therapy was coming out, and there wasn't a significant history or data that we could stand on, the observer / partner made sense. Over the years, as we've grown the therapy and we've learned more about the therapy, we always believed that we wanted to progress the indication, and the next logical step for us was pursuing a nocturnal indication, which is where a patient would, again, dialyze in a home environment, but this time during night hours with an observer present, but not necessarily observing the treatment, but more or less a partner present. Again, we were able to build mitigations and labeling and run a clinical study to support that indication to show that the therapy was safe and effective. Once nocturnal had cleared, the solo indication became important because we realized requiring our partner started to create a patient and partner burnout, and we realized that the therapy was not going to grow if we didn't start to advance not only our thinking but our indication. So when we come back to the slide and on my first bullet, we've known for a while clinically that there's a significant amount of clinical and quality of life benefits to home hemodialysis as a modality, especially when we use the modality more frequently and try to really model normal kidney function. For those that don't understand or have not been close to dialysis, it's usually an in-center treatment, usually 3 days a week, with a significant period of time, 2 to 3 days over a weekend, where you're able to dialyze again. And in that moment, a serious amount of fluid build up with the patient, and significant amount of, I wouldn't say side effects, but feeling not up to par. So we've always believed that mimicking the kidney and treating as much as possible will provide a better outcome for the patient. Unfortunately, there's been a lower penetration of home hemodialysis in the United States. Of all dialysis treatments, 1.7% in the U.S. are actually home hemodialysis. And as we've spoken with patients over the years and been part of patient advocate groups, we've realized that in some cases we have patients who are just unable to identify a partner. And because they cannot find a partner, whether it's a family member or friend, they've not been able to come on to home hemodialysis. Likewise, we have patients who lose a partner for a variety of circumstances, or the partner becomes unwilling and they have to stop the home therapy and again we return to a dialysis clinic. We've often felt that the HHD home hemodialysis partner standard is a high bar compared to other life-sustaining therapies. With peritoneal dialysis, a 2015 survey came out where 30% of those performing PD did not have a partner and 12% lived alone. So we were really trying to look at can we sort of level the bar across both therapies. And then outside the U.S., solo home hemodialysis is actually very well accepted as a standard of care. In a lot of places, people start with solo, and if the solo complications are too much of a bar for the patient, they then come in center. In the United States, it's sort of the opposite. And then last but not least, we've had a number of patient groups that have been vocal advocates of patients and being able to make their own informed choice of whether to perform solo or not. So that's been really the history for us, and then I'll get into how we started to bridge this with patient preference. Next slide, please. So one of the things about the patient preference that we really wanted to answer, we really wanted to start with what's sort of our hypothesis for patient preference, what do we want to learn? And I think we really want to understand, is our technology at enough of a state of the art to a point where now a patient would look at any residual risk in a solo HHD environment, and would they choose solo over going in center if they had an accurate representation of the benefits and risks? That was the number one thing we wanted to understand. And as we started to work with the renal branch and work over time of developing this pathway, it became really clear about what are the risk tolerance thresholds. We started out really qualitatively, would you do solo or not, yes or no. But as we migrated to more of a quantitative method in partnership with the FDA renal branch team, we realized the risk tolerance thresholds would give us something much more quantitative to say, for this type of risk, this is the percent or the statistical mean of what a patient would prefer from a risk tolerance perspective. So that was another thing we're trying to understand. The other thing we didn't know qualitatively is we didn't really understand among a surveyed patient group, what is the current use and who's out there performing without a partner, and then what were the attitudes from a qualitative perspective and also the perceived risks. And I'll get into how the pathway evolved and how we incorporated this. And the last point, my fourth bullet, probably the most critical, we often debated internally about how far do you go with the technology, and we would often debate that if we tried to have the technology mitigate every risk, we were concerned that we could be pushing something so complex as a system in a home environment that we may struggle to even maintain a solid usability of the device. So we felt we were at the technology state of the art in mitigating the risk, and we knew there was some that we really struggled if we could even mitigate, and that's what sort of brought us to that cusp of maybe now we're at a point that our risk approach from our own design perspective and our design risk assessment processes, it was not incorporating a patient, it was incorporating a far as possible worst case scenario inside the company. Next slide. So, I'd love to be able to say that we sat down in an hour-long meeting with the Food and Drug Administration that we came up with a patient preference pathway and we filed a 510K quickly thereafter. It wasn't quite as quickly and smooth as that. However, what I felt, since we were one of the first ones to do this as far as developing our own survey and working with the renal branch on the pathway, we sort of went through our own evolution. You know, we started out, if you look at the bullet points, we started out looking at our own internal risk files and really seeing what mitigations we would need and where was our state-of-the-art hypothetically. We were originally going to submit a 510K for the indication expansion and really just include supplemental labeling. But as we get into FDA discussions, we started out, if you look at the top row, you know, we started out on an exploratory informal discussion standpoint at a patient preference workshop down with FDA in Washington. And our first thought collectively between both groups was, let's maybe look at a qualitative survey and see what we learn. And as we did that, we found that there wasn't enough rigor on measure of risk. It was more just direct, more gut feel, anecdotal preference. And so the qualitative survey was a good learning for us, but it definitely taught both sides, both FDA and NxStage, that we need something a little bit more statistical rigor. And in that same process, we were debating, you know, should we provide an informed consent tool out to patients so they could basically say, I understand the risk and I agree with the risk, I'm going to move forward. And that was a discussion where we didn't think we had the regulatory mechanism to do that, so we switched it to what we call really a discussion guide. And then finally, the last part of the progression of the pathway was really working on a discrete choice model where we could really use a threshold technique and tease out the statistical preference and risk thresholds across different groups. And so when I look at the third bullet, the two biggest things I'd take away from our pathway evolution was we debated direct preference method of surveying versus discrete choice in our tool usage. We agreed on using both. However, we agreed that the discrete choice was much more quick for clearance, and we felt the direct preference was much better for marketing. And so we learned some good things in direct preference around how often do partners treat part of the time alone, or how often a partner is not there to observe treatment, what's your overall preference, how many people actually today perform solo. That was nice from a direct preference, but the discrete choice really nailed down, hey, I'm willing to take this much risk against the in-center therapy. We also collaborated on this idea of labeling. We originally thought that informed consent all made us feel pretty good, that it was more binding. But again, we didn't feel that there was a really solid regulatory mechanism, especially in the 510K process, to say this is informed consent. What we agreed on that's been really effective so far has been really a decision support tool that we supplemented our labeling with that was meant to really guide patients and clinicians on risk and making that a discussion and a collaborative decision on, is this the right thing? We do feel that solo HHD may not be for every patient, and our survey did indicate that. I think by having everything presented very clearly, you know, we're allowing the clinician, the patient to make an informed decision. Next slide. So really what we did to really nail down, you know, what we did collaboratively between the renal branch and NxStage to really have a proposed pathway is, if you look on the slide, we see the, you know, patient preference survey results were one piece. The shared decision between the patient and clinician versus, you know, via supplemental user guide was a big piece. And then we also had the same risk mitigations that were used in our nocturnal indication. There were some leak detection devices and other ancillaries and equipment and training that would also be applied to solo. Combining all that allowed us to go with 510K clearance during waking hours using the NxStage System 1. One thing that's important to know, and one thing I'm glad that we've come on this pathway, is we felt very confident going into these discussions that we've had at the time, over 14 million home hemodialysis treatments. So we knew about our device, we knew the risks, we felt we knew our patient population pretty well. We also had a previously cleared nocturnal study, which was moving that indication, as I mentioned before, sort of progressing the indication to removing the partner requirement. And then we had a history since 2005 of complaint handling and reporting. So we felt pretty informed, and I think in this pathway it was a very nice progression based on a lot of experience, both on the renal branch side and NxStage. Next slide, please. So what were the challenges and lessons learned from a NxStage, sponsor perspective? We're a small to mid-sized company. We thought we all knew statistics pretty well. We learned that we didn't have the market research and statistical expertise, and so we engaged an outside partner to help us. And that was extremely effective because they could really speak the language on different types of tools to use, and also really how to make the survey as understandable to a patient as possible. I mean, we thought that this was pretty easy to do, but when we really got into it, it was nice to have a partner, and we felt that partner could really help prepare us for our discussions with the FDA team as well. I mentioned before, the second bullet is really understanding your technology patient risk assessment trade-off. If you're looking at your device and saying, have we gone as far as possible from a risk management term, and is the residual risk really up to the patient, or is there more we can do to mitigate risk? We felt we were getting to that technological state-of-the-art for solo during waking hours, and we felt that this was a good progression for us to make from a risk perspective. One thing we learned in the survey that is really determine the complexity of what you want to put out for a survey, and really understand what's going to be reviewed by the patient, and is it going to be something they actually understand. So again, we started with a limited qualitative assessment. We found that we had a very binary population, and at first I was really excited about the qualitative assessment because it was much less burdensome to pull off. But when we got the results, we realized in a pilot that the patients were very binary in their responses. It was either I like this therapy, I don't, and there was no way to measure how far they would appreciate the risk trade-offs. So I felt the discrete choice was a good balance. The one thing, a lesson learned is the qualitative assessment, although easy to administer and easy to understand by the patient, is much more limited from an information perspective for making decisions around a regulatory clearance. The discrete choice, much more effective for clearance, but the risk is make sure that it can be understood by the patient. One of the things that we learned was start with a pilot, and the studies themselves, the patient preference study certainly is not as cost intensive and labor intensive as running a clinical study. It still is not a easy slam dunk. For us as NxStage, there was still a good amount of investment in it, and so we felt strongly about have a good pilot and understand what you're getting into, and understand sort of your patient's preference before you start going there with a high-powered statistical survey. I mean, we felt that some of our patients that we had spoken to in a pilot, again, I mentioned before, were very binary. They either were all in or not, and we were struggling with how is that going to convert to discrete choice model and threshold techniques. So that was a big thing. And then I'd say last but not least, as we get into our threshold technique and discrete choice model, we really partnered with the FDA, especially Martin Howe is going to speak in a few here, but how can we present this survey in a way that a patient can understand it and make good provide good risk-based feedback. And there were some nice models that were provided by the FDA team of how to pictorially show the risk of what you're comparing between in-center dialysis and home hemo, and that was a huge win for us. Next slide, please. And last slide for me is really what would be the advice from a process perspective that I'd recommend to other sponsors. One of the things I'd say if we had to do it over again is know going in that there's going to be some type of scientific elicitation method for the survey. It's not going to be qualitative or light, that it needs to be pretty statistically valid. So have the expertise, allow the time, and allow the time for the dialogue with the renal branch or whatever branch you're going to be working with to get that done. Know your audience. If you're going back to the old write it right type of labeling for FDA, think of a survey and think if you have a population of people that are, an 8th grade level of education on average, and you're writing to that level, trying to introduce risk-based survey statistical techniques, present it as simple as you can. That was something that we learned quite a bit as we were going through it. I mentioned before, make sure you understand your device risk before you get in front of the FDA team, and are you at a state-of-the-art or at a point, or what I'd say an entitlement level where if you went any further than this, you start to trade off device complexity and other issues. Mention the pilot, really seek the patient feedback prior to the survey. When we did that, we understood their qualitative risk, which was good to help us design the survey. We also could detect how well they comprehended what we were asking, and we started to get into potential demographic differences. One thing we learned that as we started this process, we were going to survey patients at a year on therapy only, and we realized that there was ways qualitatively from the pilot that we ought to open this up to all patients demographically that have been trained on the NxStage System 1. Goes without saying, involve the FDA branch and the Quantitative Innovation Program up front. We learned a lot of this. It was a phenomenal effort and a great project to be on as we're all trying to work through a sort of a new innovative pathway together. And then I'd say last but not least, try to administer the survey to as wide of a demographic within your indication as possible. Again, I mentioned before about how we were looking at only patients on treatment for a year. If you narrow that survey distribution, there's a good risk, there's a good chance that will come back and limit the labeling. So because we open this up to anyone that had been trained on the NxStage System 1, we felt that we're going to take a bigger risk statistically to see what may come out in the survey and maybe have a less experienced response. But what we got was a very accurate response across our demographics, so I'd highly recommend that as well. I believe we're going to have questions at the end, so I'm looking forward to any feedback or questions. I think I'm going to hand it off now to Stephanie for the next portion of our discussion.
Great. Thanks, Todd. And yeah, as Todd mentioned, if you have any questions, please submit those via the chat box, and we'll get to those at the end. But now it's my pleasure to turn things over to Frank Hurst, the medical officer in the renal devices branch, and he's going to share his perspective a bit from the reviewer standpoint of receiving and working with one of these patient preference surveys. So, Frank?
Oh, thank you, Stephanie, and thank you, Todd, for the introduction to this topic as well. So my presentation is called Use of Patient Preference Information to Expand Home Hemodialysis Labeling. Like Stephanie mentioned, I'm the medical officer in the review branch that reviewed this application. I also did want to point out that I'm just sort of one member of a team. We certainly have we had 2 engineering lead reviewers on the project, as well as branch and division management that helped usher this along. Next slide. So in terms of what I'm going to talk about, initially I guess I want to talk about why a patient preference study made sense, but before I get into those details, I wanted to also have some clinical and regulatory background on the topic. Then that's what FDA wanted to see and what we learned from the process. Next slide. So to start with some background of why patient preferences are important in end-stage renal disease, and sort of why this should be considered a preference-sensitive area. So for those not familiar with hemodialysis, Todd touched on some of this already, but it's a blood purification therapy where blood is removed from the body, passed through a filter, and then returned to a patient. And most patients with end-stage renal disease are on hemodialysis in clinic setting. And so hemodialysis is life-saving, but I think most would consider this therapy to be inconvenient. For 3 days out of the week, patients spend a fair amount of time traveling, waiting for treatment, treating, and then recovering. And then for some patients, the recovery even continues even after they've returned home. Additionally, the 3 times a week schedule makes it difficult to work, difficult to travel, and then you also have fairly intense dietary and fluid restrictions that accompany that therapy. And so there's been, a push over the past several years to increase the convenience of therapy. And again, one of the options to make it more convenient is to move the therapy into the home setting. However, as one can imagine, changing the care environment like this involves the patient making certain trade-offs, which makes the choices preference sensitive. So next slide. And so this slide shows that that patients may, or that therapies may even become more preference sensitive over time and in the future as treatments become even more portable. So this could represent a logical progression of going from the clinic, to the home to potentially an even more ambulatory therapy. The picture on the left is a typical in-clinic hemodialysis treatment. The middle picture shows the NxStage system, which in this picture is sort of a tabletop system that can be used in the home. And the last image is a concept for a wearable hemodialysis system, which these are not currently available, but there are some that are in development. And as you can imagine, going across the spectrum, there's a variety of trade-offs that patients are going to have to make. For example, if the system is a wearable system, there may be certain benefits associated with that, but it could also impact the patient's ability to shower or exercise, or work for that matter, depending on what their job is. Next slide. But coming back to home hemodialysis, which is the topic for today's discussion, this slide gives some examples of trade-offs that patients have to make before moving from the clinic into the home setting. So on the left, on the plus side, there's increased flexibility, both to work and travel, and then also more flexibility with the therapy options. So Todd had mentioned, nocturnal therapy, but there's also sort of extended therapy and frequent therapy, which, these types of therapies could improve the overall quality of life because they more closely mimic native kidney function, which, the native kidneys work 24 hours a day, 7 days a week. The column on the right lists some of the negative aspects of bringing the therapy home. So along with the need for dedicated space, there may be an increased burden on family members, as well as the increased responsibility for the patient and their care partners. And for the increased responsibility, that includes performing all aspects of the treatment yourself, as well as managing all the risks associated with the treatment. And of note, there are no trained medical personnel in the home during the treatment, which could present a problem, if and when issues may occur. Next slide. And I just wanted to highlight one of the issues that could occur that FDA was concerned about throughout this process, and that's a venous needle dislodgement or venous needle disconnect. And so typically for a hemodialysis treatment, you use 2 needles for blood access. 1 needle withdraws the blood from the patient, and 1 needle returns the blood to the patient. And so if the venous needle becomes dislodged, it can lead to rapid blood loss, and patients can actually bleed from both the needle site and from the machine circuit. And if you can imagine, if you have a large enough amount of blood loss, the patient can actually lose consciousness, which could impair their ability to respond to an event like this. So if this were to happen, they may not be able to turn off the machine or call for help. And if this were to happen, I guess the hope and sort of the FDA background is that there would be a care partner there that could be their safety net. Unfortunately, these events are rare, but they do occur and have severe consequences. I have the frequency listed on the slide as well. There's some data that show that in clinic these happen, roughly about once every 3 to 5 years. The frequency is less known in the home population, but the presumption is that it's roughly the same. Next slide. And Todd touched on a lot of this, but in terms of the regulatory background for the care partner requirement. So in 2005, FDA cleared the first system for home use, which allowed for the convenience and flexibility of dialyzing in the home setting. But because of the lack of trained medical personnel, FDA wanted to mitigate some of the potential additional risks that could occur. So as part of the labeling, FDA required that all treatments must be observed by a trained and qualified person considered to be competent in the use of the device by the prescribing physician. And so, fortunately, with the 2005 clearance, it expanded treatment options for many patients Unfortunately, for the patients that did not have a care partner, they couldn't have home hemodialysis treatment as a therapy option. And because of this, FDA and industry had many discussions on the care partner requirement, but couldn't really come to a consensus given the lack of data. And based on the frequency of occurrences, it'd be really challenging to do a traditional clinical trial to evaluate these rare but serious events. And FDA was concerned that removing the care partner would increase the risk to the patients, and we didn't believe that patients would be willing to accept this additional risk. Next slide. And so this issue, I wouldn't say it was dormant for 10 years, but it resurfaced at a 2015 workshop that Todd also mentioned, where many topics related to patient preferences were discussed. And so there were a variety of stakeholders at this workshop, including patients, and the discussion revolved around involving patients in the device development process to bring new technologies to market and improve existing technologies. And in addition to improving technologies, some thought it would also be important to expand access to therapies that were already available, thinking that many were underutilized. And so during the workshop, industry, FDA, and patients kind of revived the care partner discussion and considered whether or not it would be possible to use patient preference data to bypass the current roadblock. Next slide. So the question posed was whether or not patient preference data could be used to expand the labeling for home hemodialysis to include patients without a care partner. Next slide. And so this is the slide's called What We Wanted to See, and Todd kind of touched on this too. So honestly, in the beginning, I don't think we were quite sure what we wanted to see. I think we wanted to, have some quantitative data, and we wanted to feel good about the direction. But this is a relatively new area for FDA, and as Todd mentioned, this type of data had never been used for a device cleared using the 510K pathway. But we had several meetings and a fair amount of discussion, and we ultimately decided that a well-designed, robust patient preference study could potentially alleviate or at least address some of the concerns that FDA staff had. The data would need to be quantitative and would need to inform FDA what level of risk the patients would be willing to tolerate in order to have the option to do dialysis at home without a care partner. And additionally, we wanted the maximum level of risks that the patient would be willing to take. The MAR would need to, we wanted to see that this exceeded the likely frequency of these events, ideally with an adequate safety margin. We also wanted labeling that adequately informed the patients of the risks, and then also device safety mitigations that considered the modified care environment. Next slide. And so this slide is what we learned from the process. I think first we learned that this group of patients had a much higher risk tolerance than we initially thought. And we wouldn't have known this information if the patients had not been asked. So it's important to keep them involved in the development process. Second, we acknowledge that this is a relatively new area for both FDA and industry, but we hope to build on these early successes to improve the use of these types of data in the future. And we do believe that the use of these data could be a solution for similar situations where there might be a roadblock. Third, we learned the importance of device labeling when pursuing an expanded indication based on preference data. And I think it's important to highlight the preference data in the labeling, but it's also important not to forget that each patient is unique in terms of risk tolerance. So even if most patients or the majority of patients would be willing to accept certain risks, the therapy still needs to be individualized after a detailed discussion of the risks and benefits. In other words, I think it's important that patients have access to a therapy, but we also want to ensure that the expanded access serves the appropriate patient population. And certainly identifying that population can be challenging. And finally, and perhaps most important, is that FDA and industry can work together for the mutual benefit of patients. Next slide. And so this is my last slide. This is a quote from a patient that was emailed to the FDA review team after the public notification of the clearance for the new indication. And so the FDA doesn't always get this type of praise, but I think the quote emphasizes what expanded access can mean to patients. And so again, that's my last slide. I'll certainly be happy to entertain any questions at the end.
Great, thank you, Frank. And now we'll pass the baton over here to Martin Howe, who is the Associate Director for Quantitative Innovations at CDRH, and really has been a leader within CDRH in advancing the use of patient preference information. Martin?
Thank you, Stephanie. And I also want to thank Todd for his excellent presentation about the sponsor's perspective. And I would love to hear more of that from other people who are interested in using patient preferences for their regulatory application. And I find Frank's presentation about the clinical aspect and the regulatory background very useful for me to understand. And as we have heard from both of them, that it is a very beginning of this stage of hearing patients systematically and scientifically, there are some learning curves for us to overcome. But I wanted to say that our center and my program are very committed to work with individual sponsors and to figure out what are the regulatory questions in hand and how we can address it innovatively using patient preferences. So for the rest of the sessions, I'm going to talk about first, going to step back a little bit and take a look at what are the general role of patient preferences in regulatory decision-making at CDRH. Regardless of pre-market approval decision or IDE or compliance, reviewers and regulators at CDRH are always facing a benefit risk assessment question. And it also, when we are doing that, it has to be in a specific adequate context. So in order to lay out the factors that we consider in making those benefit risk assessments, we have released 3 guidance documents that cover the pre-market, the IDE, and the compliance policy decisions respectively. Next. So next we're going to take a look at how patient preferences are being used in these benefit risk assessment. We can see that in terms of patient input, there are 2 types of patient inputs that we regularly encounter at the FDA. The first one is the patient-reported outcome is basically reflecting the benefits and risks include effects on patients' health-related quality of life. And as for the patient preferences, it's more specific, getting into or informing us in our benefit risk considerations. Some of the information include which benefits and risks are most important to patients, and whether one set of benefit risk trade-offs are acceptable from patients with regard to their reference conditions, and whether there are clinically relevant subgroups of patients that would benefit, would choose a particular benefit risk profile over the others. And finally, does the PPI manage to capture a diverse preference across spectrum of indicated populations so that we won't be just limited by the samples that we studied. Next slide, please. So, although we have talked about patient preferences are very important and useful, we haven't, these guidances didn't talk about how to measure them. So as a result of that, we set out a patient preference initiative to answer that specific questions. First, we kick off the initiative with with obesity studies of patient preferences for weight loss devices. After we publish it, we have a joint collaboration effort with the MDIC to develop a patient preference framework report. And the report has two parts. One is that the regulatory context of patient preference information, and the second part is a catalog of existing patient preference elicitation methods. And then in parallel, we have developed a guidance document on patient preference information to further clarify our position in terms of using patient preferences. And after we have accomplished the report and the guidances, we deepened our commitment to using patient preferences in our 2016-2017 strategic priorities, which specifically stated that our center would be included more cases that consider patient perspective in our regulatory decision making. And the result of that strategic priorities would be released in a couple months. Next slide, please. So as I said, the catalog of the MDIC report is a collection of existing quantitative methods for us to measure quantitatively the patient preference information. As you see here, there are 14 methods listed, and the obesity study that I mentioned used a method called conjoint analysis and discrete choice experiment, which is a method that Todd had mentioned quite frequently in his presentation. And this was also the first method that he came in to the FDA to discuss his potential use of patient preferences for his product. But then as we work with the renal branch, our colleagues at renal branch, and the sponsor and Todd, we take out what are the research questions in hand and what are the essence of the questions. Why do we want to know that? And what can we learn in order to clarify that? So we later determined that perhaps a threshold technique would be a better way to address the questions in hand, which is because the threshold techniques fit in a situation where we have a comparator, which is the standard of care of dialysis in center, and a potential product that can offer benefits, potential benefits that are quite different from the traditional standard of care. But at the same time, we wanted to know what are the risks that would be involved that would be acceptable to patient in exchange for that benefit. So next, I'm going to dive into a little bit about threshold technique. Next slide, please. So threshold technique is basically refers to a format of questions that we ask the patients to answer. So for each patient, they will answer a series of pairwise comparison questions. In each question, the patient would choose between two treatment profiles. One is a reference and the other is alternative. And typically, a reference would be control, standard of care, or status quo of their condition. And versus the alternatives are the new devices. And a treatment profile is defined as a combination of benefit, risk, and other attributes such as the convenience or the location of the procedure. And one specific thing about threshold technique is it focuses only on one risk or one benefit of the device. And in this case, we are focusing on the mortality risk of potential mortality risk of the new device. We first ask patients to choose which one of the standard of care or the new devices would be preferable to them. And then we change the risk profile or the benefit profile of the new devices based on the patient's response in the previous question. so that we are trying to etching into the maximum risk level where they would switch their decision. So hence is the name threshold. So it's basically a way, again, to measure a patient's maximum acceptable risk of choosing a new device over the comparator. Next slide, please. Here is an example of a threshold techniques. Suppose that we have a new device that offers a novel benefit, but with a higher risk than a standard of care. So we wanted to know what kind of risk threshold that patient would have to receive that device rather than the standard of care. So we ask patients, assume that the standard of care have a 2% risk of certain events versus the new device has 10%. And if the patient chose the new device, then next we are going to make the new device less appealing or attractive to patients by lowering, by increasing or elevating the potential risk level of the new device. And then we ask the patients to choose again, is 14% versus 2%. And if the patients choose the new device again, then we would further increase the risk of the new device into 20%. However, if the patients chose the standard of care, then we would know that the threshold risk tolerance or risk level for a patient would be somewhere between 10% and 14%. However, if in the initial questions the patient had chosen standard of care, then we would make the new device more appealing, which is to lower the risk of the new devices from 10% to 6%. And if the patient chose the standard of care, then we would make the new device even more appealing by lowering its risk to 3%. However, if the patients that chose the new device at this juncture, then we would know that the patient's risk threshold risk tolerance would be between 6% and 10%. Next slide, please. So here's a list of possible sequence of the patients that would go through when they are answering this sequence of questions. And as we can see, different questions can reveal different ranges of their risk threshold. And by using a technique called interval regressions, we can not only estimate the mean risk tolerance of patients overall, but also we can estimate it by group, and even we can test statistically testing their risk tolerance across different subgroups. Next slide, please. So I would like to perhaps just step back and talk a little bit about the general steps to measure preferences. So we are not so that we can convey a wider message or lesson that we learned from this exercise. In general, despite different techniques, we still have been trying to answer one question, which is which one option would you prefer and how important it is. And there are two very important terms that we want to know. One is the attribute, is basically a description of benefit risk and other considerations, and levels within attributes. It can be categories or values. It can describe the size of benefits or the risks or the probability of the risk and vice versa. And then regarding the general steps, here is a much simplified steps to conduct a stated preference method. It involves two parts. The first part is the qualitative steps, which involve we have to first identify the attributes of interest. So these involve patients, the sponsors, clinicians, and of the FDA. It's very different from the traditional patient preference studies because we are collecting this information to answer regulatory questions versus the other preference studies are mostly interested in patients' perspective only. So during the quality step, we have to be careful and design the survey instrument and pre-test it with patients. And then afterwards, we develop experimental design and specify analysis plan and analyze them and interpret them correctly. Next slide, please. So in our patient preference information guidance, we list out our recommended quality of the studies, mostly because we want to avoid being prescriptive and stifle the development of this newly developed area, and also our application of this information in our decision has been relatively new. So therefore, we also don't want to be too restrictive in terms of what is and what is not. So therefore, we just highlight some of the high-level principles, and they are clear. They are explained very detailed in a very detailed way in our guidance. Next slide, please. So here is my list of recommendations and lessons learned from doing the patient preference studies and collaboration with the sponsors. And some of these lessons are echoes excellent and perfectly with the previous two speakers. First, talk to FDA early and often, because again, the choice of the preference attributes and levels are highly important in terms of answering the regulatory decisions that we have to make within the FDA. And we also need to choose the appropriate study design. And if there are some subgroups that we wanted to study further, then we would like to use a stratified randomization to address that. And finally, we wanted to encourage the data quality assessment plans built in the protocol. Next slide, please. In terms of the PDI documentation, we are very seriously about hearing and respect the patient's perspective in patient preference studies. Therefore, we look at the evidence showing that the patient really understands and being cognitively feasible and capable to answer these questions. And how can we do that? Is to when the sponsors are conducting their study, either they are through a pilot study or what we call a pre-test of their instrument, it should be very detailed or sufficiently detailed for us to tell the patients being able to answer them within a reasonable amount of time, which can be between 20 minutes to 45 minutes, and they also understand the individual concepts. And so, therefore, we would love to see the pre-test documents before we finalize the survey instrument. And again, we have to specify subgroup prospectively, and so that we can capture the patient's heterogeneity. And again, data quality is very important. Next slide, please. Last but not least, I would like to talk about how to submit PPI patient preference information data to the FDA. Here I list two colleagues, two email addresses. One is mine, and the other is Annie Saha. She is the center's lead on the patient preference information initiative. And if you have any questions, feel free to email me and Annie, and we would be happy to link you up with the colleagues of interest, and then we can start a conversation. It doesn't necessarily even to be specific to any device under review. So next slide, please. Finally, I would like to provide some of the additional useful information. I mean, the first one is, again, come early and often to talk with your review divisions. And here we also list the patient preference guidance. And one thing that I want to highlight is that we recently developed some of the discussion points about things to consider when you're conducting patient preferences during your qualitative steps. As is not new here on the slide, they are very carefully developed, and I hope that that can be very useful and help sponsors to consider patient preference studies. And last but not least, we are going to have a public workshop sponsored by all 3 medical centers, including devices, drugs, and biologics, in Thursday and Friday this week. And the link provided here can lead you to registration. Next slide, please. So, to summarize our presentation, I would say that we are very, we as a center are very interested in promoting considering patient preferences and perspective in our consideration of regulatory decision making, and is one of the very high prioritized initiative within our center. So the FDA blog piece that I show here provide an excellent summary of our efforts, and the regulatory impacts of using patient preference information. And the photo in the picture in the blog is our center director, and as you can tell, he's a smile. He's very happy about the progress that we made. But at the same time, he also, along with us, also wanted to extend our invitation for your collaborations and consideration of using patient preferences. And hopefully, the progress we made so far have shown substantial commitment from our center to forward the consideration of patient preferences in the future. Thank you.
Great. Thank you, Martin. We have just a few minutes remaining for any questions. Again, I encourage you to submit your questions via the chat box. I will let everyone know that we will be making the recording and the slides from this presentation available on the MDIC website, and I will make sure to send those links to everyone who is registered for today's webinar when they are available. Martin mentions these resources already, but I wanted to make sure and put these up on the screen as well. We do have MDIC has several resources related to patient preference information, as well as our other regulatory science initiatives. Also want to let you know that there is a upcoming, you can find links to other upcoming webinars on MDIC.org/MDICx. All right, I'm not seeing any questions. Oh, here we go. No, I take that back. Question regarding getting started. Should companies work with their division or the Quantitative Innovation Program to get started?
I would say both. Actually, when you are contacting your review divisions, you can more than welcome to mention my name and any SAHA's name. And specifically ask the review division to include us in your consult, so that we will be able to connect it with the review divisions to discuss the questions in hand with you.
Great, thanks, Martin. All right, well, we are just about at the end of our hour here. I know that if you do have additional questions, Martin has made his email address available so that you can contact him directly regarding, and often, apparently, regarding patient preference information. Thank you all for joining us today. As I said, we will make the video and slides available in the coming days, and go to MDIC.org to sign up for other MDICx presentations.
Thank you.
All right. Thank you, everyone.